This method can help answer key questions in the microRNA field by providing tools to detect the relative localization of both protein and microRNA molecules. The main advantage of this technique is that you can detect both protein and microRNA molecules on the same tissue section.
This protocol begins with a rehydration of the slide-mounted tissues. Warm the slides at 60 degrees Celsius for 10 minutes in the hybridization oven. The paraffin should be visibly melting. Then, incubate the slides in a glass Copland jar containing commercially available clearing agent for 10 minutes. Do this twice. Next, transfer the slides from wash to wash to rehydrate the tissues. Once hydrated, incubate the tissues in proteinase K working solution at 37 degrees Celsius for 15 minutes. To remove the proteinase K, wash the slides in 1x PBS 2 times. Now, fix the tissues. First, make reservoirs on the slides using a hydrophobic pen to draw a water-repellent circle around the preparations. Next, apply 500 microliters of 4% PFA solution within the barrier and incubate the slides in the fume hood. To remove the fixative, wash the slides in a Copland jar containing PBS for 5 minutes 2 times at room temperature. Next, wash the slides in 0.13 molar 1-methylimidazole for 10 minutes. Do this in the fume hood at room temperature and perform this wash twice. Add the EDC fixative and incubate at room temperature in a fume hood for 1 hour.